important to be able to identify a molecule that is absent in the human host but present in the parasite, and is also essential for the survival of that parasite, so that at least at the end, if you develop a compound that when it blocks the activity of that particular protein, it will kill the parasite. So generally, to produce a drug or to give or to bring drugs into the market, it took not less than 12 to 15 years, because there are a series of steps before you were able to produce a drugs. We have basic research starting from biochemists, microbiologists, and biologists. So it starts from basic research, identifying a target and the compound. Then preclinical trials, after you were able to develop or identify the compound, then you test it in a mice, in animals, both in vitro and in vivo. When we said in vitro, it means that when you are carrying out a test or an observation outside a living system, like for example, look at it here. It's like it's a blood that scientists collected from a mice. Maybe in that particular in that particular blood, there is a particular proteins or a parasite that you want to target. Maybe there is a parasite in that particular blood. So you now get the compound, purify it, and put it into this petri dish, and you now check if the compound, which is the drugs, will kill or will have effect on the parasite. So you see, you get the blood outside this animal and you carry out a test. So this kind of test, we call it in vitro. But in vivo is when you take the drugs and you inject it, or the compound, you inject it into the animal. So when you are carrying out a research inside a living organism, so we call it in vivo. When you are doing a test into an animal or into a living system, we call it in vivo. And then we have clinical studies, which comprise phase 1, phase 2, and then approval. Then there is also post-clinical trials. So it took not less than 12 to 15 years to bring drugs into the market. So we have different ways of producing drugs, but with time, because all this thing that we are doing is like a basic explanation. So with time, we are going to look at all those things. Then there is also prenatal screening. If you have a baby inside you, you can investigate whether the fetus has genetic disorders or anomalies. Like for example, if you have a baby inside you or in a pregnant woman, you can check whether the baby have sickle or not, or other like genetic diseases. Then it's also used in agriculture, in evolutionary biology, and in forensics. In forensic, you can just get the blood from, or maybe when there is a crime, and you went to the crime scene and you are able to see a blood, you can get that blood, you can isolate the DNA and identify the DNA is from which particular individual. And from there, that is why a lot of criminal cases have been solved with the forensic, that is DNA genotyping. So that is brief applications of human genome projects. And then, so ladies and gentlemen, we are now going to look at the other, or how, or other areas that biochemistry relate with. So remember, as I said, biochemistry, we are studying life using a molecules. So with a molecules, remember in a previous slide, we talked about some molecules or some carbohydrate or some proteins that are produced in order to produce or to make a particular cell to produce a specific structure, like your head, like your brain, like eyes. Is all the structure is as a result of a proteins that produce from the DNA, which is biochemistry. So biochemistry is important in knowing the anatomical structure of a living organism, in knowing the plant, how the plant look like, the reactions that are taking place in that particular plant. If the plant develop a particular disease conditions, how scientists or how biochemists will be able to identify the problem and then also bring the solution of the problem. And then pharmacy, that is drugs. So I just finished explaining how biochemists can be able to produce a drug. So biochemist is also related to pharmacy and also nursing, that is in disease treatment and diagnosis, and also microbiology as well as a zoology. So biochemists, what biochemists usually does, they can isolate, purify, characterize, and study the reactions of all cellular. So therefore, what I want to say now, biochemists can isolate, can purify, and characterize a particular molecules from a cell. And we also have other branches of biochemistry. We have toxicology. Toxicology includes the study of the drugs and other xenobiotics with their effects. We know that generally drugs, they are xenobiotics because they are foreign and they have their own biological effects. So generally drugs have side effects. So the study of the drugs and their side effects is called toxicology. It's called toxicology. And then you also have lipids and carbohydrates biochemistry. Some diseases like cardiovascular disease, diabetes mellitus, obesity are as a result of accumulation of lipid and some carbohydrate. So lipid and carbohydrate biochemistry give us an idea of how lipids and carbohydrates lead to disease conditions like the atherosclerosis, the cardiovascular disease like stroke, coronary heart diseases, and others. Because all these diseases, lipid and carbohydrates, are associated with diabetes mellitus, obesity, and others. Then we have natural product biochemistry. Some natural products or some products produced from plant are used in drugs like quinine and artesunates. Artesunates and quinine are the drugs that actually produce or that are isolated from plants. So the use of compound from plant in treatment or in designing a particular, yeah, especially in the use of compound from a plant in the treatment of diseases is known as a natural product biochemistry. And in fact, some natural products from plant can also use in a lot of other things like in cosmetics. And then we also have the real branches of biochemistry. These are the branches of biochemistry apart from toxicology, lipid and carbohydrate biochemistry, natural product biochemistry, that is enzymology, metabolism, plant biochemistry, molecular biology, animal biochemistry, cell biology, immunity, and genetics. They are all branches of biochemistry. And with time, as we are doing this course, we'll get to understand each and every one of these. Okay? Then now, next slide. So we are now going to start discussing on cell. What is a cell? A cell is the basic living structural and the functional unit of living organism. So a cell is life. If there is no cell, there is no life. And the first scientist that discovered cell is Robert Hooke in the year 1865. Is the scientist that was able to discover cell. And these cells, they are responsible for all the activities of a living organism. In a cell, we have different organelles, which include nucleus, mitochondria. Mitochondria is responsible for producing energy in a cell. So if you remove the mitochondria from a cell, that cell will not produce energy and the cell will die. So any disease we have is coming directly from a cell. If you have liver problem, it's because you have problem with the myocytes. That is the cells of the liver. If you have problem with your brain, like you have psychiatric problem, then the problem is also coming from the neurocytes, the cells from the neurons. And then if you have diabetics problem, cardiovascular disease, also the problem is coming from a cell, because the cell is what form a tissue, and the tissue form an organs. So if you have problem with a particular tissue or with a particular cell, of course, you will have problem with that particular tissue. So if you have a problem with a myocytes, that is the cells of the muscles, you will have problem with the muscles. So that is why even under a cell, we have what we call cellular injury. So under cellular injury, remember the word injury is when there is alteration in a normal function of a particular system. So you said a cellular injury. A cellular injury is when there is an alteration in a function or on a function of a particular cell. And we have two types of cellular injury. We have reversible cell injury and we have irreversible cell injury. So if you have a disease or if you develop a disease and you were able to cure that particular disease, so that type of injury is called reversible injury, because you were able to reverse the normal function of a particular cell. And that is why you were body cells or that particular cell now restore its normal function. Restored its normal function. So a cellular injury, it can be reversible or irreversible. So if the injury cannot be reversed, then the cell will die. So under cellular death, we have apoptosis and necrosis. So what is the difference between this necrosis and apoptosis? Apoptosis, apoptosis is a programmed cell death. It's your body that program that after so number of time, or after so number of minutes, or after so number of days, this cell should die. Like for example, our red blood cell die after 4 months, that is 120 days. So it is programmed. And we also have some cells that they are injury or they develop or they have an injury, and we don't want those cells to further divide, then we eliminate them with apoptosis. And as I said, we have necrosis. So when we said necrosis, it means that it is not programmed and it is pathologic. It's pathologic. So whenever you have the death of a cell as a result of necrosis, it means that there will be disease. And in human, there are, or in average human being, there are like 32.7 trillion cells. In average human being, there are about 37.2 trillion cells. And also have like over 200 different cell types in our body. And these 200 different cell types, we have like the cells of the muscle, the cells of the brain, the cells of the kidneys, the cells of the tongues, the cells of the eyes, the cells of the kidney, So all those kinds of cells, we have different types of them. So we have 200 different cell types, different cells. But if you are summing the whole of the cell, in average human being, we have 32.7 trillion cells. So all cells produce, all cells produce them by a process of cell division. Remember, in cell division, we have meiosis and mitosis. So a zygote cells, which is produced as a result of fertilization of sperm and the egg cells, are produced through meiosis. So meiosis is taking place only in sex cell, which is sperm and egg cell, while the mitosis is the process whereby a cell divides itself into two, and it's usually taking place in somatic cell. The somatic cell is called non-reproductive cell. So cell biology is the study of structure and function. The structure of cell is intimately related to its function. So some cells have different structure, and that different structure is what makes them to have different function. So we have different parts of cell. We have plasma membrane, we have cytoplasm, and then nucleus. So cytoplasm is made up of cytosol and organelles. So these are some organelles of these cells, and we are going to look at them one after the other, their functions. So ladies and gentlemen, can we stop here or we should continue? Let me hear from you.
We should stop here, sir.
We should stop here and continue, sir.
Let's continue, sir. Let's continue.
Tomorrow.
Let's stop here and continue tomorrow, sir. Thank you.
Let's continue, sir.
Okay, I will continue.
Yes, sir.
Yes,
Continue.
Continue.
Continue, sir.
Cannot digest
Continue, sir.
So these are the different components of cells, and we are going to look at their function one after the other with time. So as I said, we have plasma membrane, we have cytoplasm, and then nucleus. So now we are starting with the plasma membrane. We are starting with the plasma membrane. So what is the structure of plasma membrane? The plasma or the membrane, that is the cell membrane, is composed. So if what constitutes or the composition of the plasma membrane, they are made up of two basic molecules. They are made up of two macromolecules. So what and what that make or that build this plasma membrane? So the membrane of the cell is made up of proteins and lipids. So if you have proteins and lipids, most of the lipids that are found in the plasma membrane, we call them phospholipids. Call them phospholipids. It means when we said phospholipid, it means lipid and phosphates. So membrane is made up of two major components, proteins and lipids. And specifically, which types of the lipids is phospholipids. And it binds together by non-covalent forces. So these proteins and lipids, they are bind together by non-covalent forces. What are the examples of non-covalent forces? I hope you are jotting all these things. So example of non-covalent forces, we have hydrogen bonding, we have van der Waals force of attraction. It's an example of non-covalent forces. So the phospholipid molecules have heads, which is electrically charged and hydrophilic in nature. A cell which has no charge and hydrophobic in nature. So ladies and gentlemen, a lipids a lipid, generally lipids, they are hydrophobic. That is, it means they have phobia with water. Hydro means water. Phobic means phobia. So lipid is hydrophobic in nature. That means it doesn't like to interact with water molecule. It doesn't want to interact with water molecule. It doesn't want to interact with water molecule. So we call those kinds of lipids called hydrophobic lipids. And we have hydrophilic. When we say hydrophilic, when we say something is hydrophilic in nature, it means that it like interacting with water molecule. It's like always to interact with water molecule. So phospholipids is an example of lipids that have both hydrophilic and hydrophobic nature. It means that some part of it interact with water, while some part of it doesn't interact with water. And in this layer, the sugar molecule is embedded in between them. So remember, I said membrane is made up of carbohydrates and proteins. It's made up of proteins and lipids. So in between these proteins and lipids, or in between this membrane, there is a sugar molecule that's embedded. So this is a typical examples of how the membrane look like. You see, this is a carbohydrate, so it's made up of carbohydrates. We have glycoprotein. And what is glycoproteins? A glycoproteins is a proteins that have both. It's a proteins that have both protein and sugar. So glyco is sugar and protein. So glycoprotein is a combination of sugar, which is carbohydrate, and proteins, and glycolipids, sugar and lipids. And then we have globular protein, integral protein, and peripheral proteins. Integral proteins are the protein that fully embedded into the membrane. They are fully embedded into the membrane of a cell. While peripheral proteins, they are just peripherally attached. They are just transiently attached to the membrane. And what are the functions of this membrane? They provide a protection between the cell. They provide protection between the cell and the outside environment. The outside environment.